because my first motivation was to do essentially cancer immunology, or more general, I wanted to do something against cancer. And this was very early, actually before I started my studies. That's how I said it's very strange. This is a long time ago, when I was about 20 years old. I did civil service right after school, essentially, and then I had to work on a hospital, in a cancer station, actually, in a cancer ward, actually in a radio oncology ward. And then I saw many people with cancer, many patients with cancer, and I saw many of them dying. And this was, of course, I was very young, was very impressive for me. And I saw all the doctors trying to treat the patients, and they were not successful, at least not completely successful, because the patients died anyway. And during this time, I changed my plans for the future. I wanted to be a technical engineer, actually, and then I changed my mind, and I thought I'd do something against cancer. And I didn't know what to do, in which direction, but I was sure I had to do something on natural science, basic science, and did not want to study medicine, because I saw the doctors being not successful in the fight against cancer. So I thought I should do something basic in basic science, and I wanted to learn essentially biology and biochemistry in order to try to develop something against cancer. I didn't know what it was. And this was actually the start, and I started actually biology at the University of Tübingen to study biology. And during this study, I learned something about immunology. We had a journal club at the university there, and there we read a very influential paper by Rolf Zinkernagel and Peter Doherty. And this paper showed for the first time the MHC restriction of T cells. This means a T cell, which can kill virus-infected cells at that time, as was it known. These T cells recognize something from the virus and something from the infected host cells, and this something was the MHC. And this was described in this famous Nature paper, actually from, I think, 1984. And then I thought, well, if these T cells can recognize something on virus-infected cells and kill these virus-infected cells, maybe this is also true for cancer. This was a speculation, and there was not much known about cancer-specific T cells before. There were some hints and some reports, but it was not clear what they might recognize such T cells. And so this was essentially my decision then, as a very young student of biology and biochemistry, to go to learn immunology, to find out whether it's possible to do something against cancer. And this was essentially, this is the answer to your question. I first became interested in cancer vaccines. This was a follower of this, actually, in cancer immunology. And based on this, later this turned into my interest in personalized cancer vaccines. Obstacles are the same as since 30 years or more. It is very difficult to bring revelations from basic science, also from animal experiments, to clinical practice. So we know in the meantime that cancers express antigens or can be recognized by the immune system, by the adaptive immune system, by T cells essentially, and that these can be translated into cancer immunity. At least this has been shown in mice and also has been shown in a few cases in humans. And the biggest obstacle is still to bring such ideas into useful clinical trials. It's not enough to show in vitro that the T cells can recognize cancer. It's also not enough to show this in mice. The biggest obstacle is to bring these observations into clinical practice. And there the obstacles are the sheer costs. It's difficult to finance such studies, especially if it's a new approach where the success has not been shown so far. It's very difficult to find enough investment to finance such clinical studies. So I think this is a basic obstacle. And one example is cancer vaccination, where I work since many years, as you had mentioned before. The other problem is that cancer vaccination has been tried certainly since 30 years or more, and it never had been successful, essentially. At least not in the sense that an improved therapy is based on cancer vaccination. This is changing right now, I think, but still, so far, with one little exception, there was no success in cancer vaccination. And so if my team and I, if we approach investment companies or actually pharmaceutical companies and say we want to do a cancer vaccination trial and we now know how to do it, we have the right antigens and good adjuvants, but then the typical reaction is, well, cancer vaccination did not work in the last decades. It will not work in the future. We will not invest into this. So this is one example of the failure, of the problems we have in the development of cancer immunology. This is that we identified the rules MHC molecules present peptides to T cells. So as I mentioned before, virus-specific T cells recognize something from the virus on MHC molecules. And in the meantime, it's known that something from the virus is a peptide, which is presented on MHC molecules. And when this was known, that virus-infected T cells present peptides on MHC molecules, we tried out to find such peptides, not only from virus-infected cells, but in general. And so we worked on with my little team when I was a young independent investigator with a small group. We tried to elute the peptides directly from the MHC molecules from infected cells or from normal cells. And this achievement had been, which I was after for several years, had then been achieved successfully by two students of mine, Kirsten Falk and Olaf Rötschke, in my lab at that time at the Max Planck Institute for Biology in Tübingen. And so we were able to elute the peptides from MHC molecules and also to identify their sequence. And the most important discovery was from our small team at that time was that we found the rules of MHC molecules, how they present peptides, the so-called MHC motifs, or for human would be called HLA motifs. So this, together with the procedure to isolate such peptides, made it possible to identify the peptides presented by MHC molecules also from cancer cells, and to predict the peptides presented from known protein sequences on MHC molecules. And this opened the possibility to for deep cancer immunology research. So our biggest breakthrough was to find the so-called immunological code, the HLA MHC motifs of peptides, which enabled then the finding of many antigens of interest from cancer antigens and from infectious agents, and also essentially started the possibility to really predict naturally presented peptides, which was essentially the start of bioinformatics with regard to HLA presented peptides, and which is also the basis to predict the neoantigens, for example, in cancer, which is now being used widely. So this is the finding of the HLA motifs was I think our biggest breakthrough in general immunology, with a particular importance for cancer immunology. And actually my first mentor, Jan Klein, a very famous immunogeneticist, where I did my diploma thesis and also my doctoral thesis, he said, when I approached him and asked to supervise my work, I told him about my motivation. I want to do something against cancer. Then he laughed and said, the immune system will never be able to do something against cancer. You are losing your time, do something else. So he very much discouraged me to go into cancer immunology. But I did it anyway. But I'm very thankful to Jan Klein, my first mentor, that I could learn from him in an excellent way immunogenetics and basic immunology. So he was against, he didn't think that cancer immunology will work, but he was a great mentor for me to do basic science. And there I learned actually the relationship between MHC molecules and T-cell recognition. And actually Jan Klein also did not believe that MHC molecules present peptides, but I tried to find such peptides anyway in his department, actually in a small group in his department. So I'm very thankful to him, and he was a very great mentor. And then my next mentor was my host of my postdoctoral position, Mike Bevan. This was in La Jolla in California. And there I learned a lot about basic immunology, and I also learned he taught me how to design experiments in a way that they give results, even if the experiments are negative or did not fulfill the expectation. Other experiments should be designed in such a way that they have a hypothesis which can be proven or disproven. And this I learned with Mike Bevan. And also basics about T-cell immunology and MHC restriction. And in particular, I learned from him how to analyze the T cells, recognizing minor H antigens. Minor H antigens is something which is similar to tumor antigens, but these are polymorphic normal antigens with some features similar to tumor antigens. So Jan Klein and Mike Bevan are my great mentors in basic immunology, T-cell immunology and immunogenetics, but not in tumor immunology, because both didn't think highly from the immune system being able to do something against cancer. This has changed now, certainly. After checkpoint inhibition had been developed, then it was clear that the immune system is very efficient to do something against spontaneous human cancer. The advice to young scholars, I think, so my advice would be follow your own ideas, or follow your own motivation, which in my case was to do something against cancer. And also for the present students, follow your motivation and your own thoughts, and don't listen too much to your mentors. Have your own thoughts and try to push your own thoughts. And of course, honor your mentors, because they are very important for you to learn the basics. Yes, I was attracted, of course, to the journal because I'm both interested in cancer research, cancer immunology, and I'm also interested in aging, in senescence of immune cells, and also of in aging of patients. So, of course, it's clear that old patients do not respond as well to any kind of immune challenge as compared to young patients. This is in particular true for people with chronic virus infection like CMV infection. They are they have this improvement of the immune system. They cannot respond as well as younger people. And another reason for joining the journal was that I know the editor very well, Graham Parridge, and he convinced me that this is a great journal, and so I joined it. And suggestions for the journal for the further development: go on as you started. Try to attract people writing papers for them for the journal. Of course, in particular, famous immunologists or famous cancer immunologists. But I know from my own experience, it's difficult to find scientists to contribute an invited manuscript review, because there are so many requests for such things that usually the answer will be negative, as you probably know from many examples. But it's good for the journal to specialize in the connection of aging immune system and cancer immunity, because most, not all, but most cancer patients are of older age, and they already appear to have problems with their immune response.